Okay, so this PowerPoint is going to be Chapter 10, Section 2. So we're on the last section that we're going to cover here. We're still talking about cell division, specific type mitosis. And in this part of the chapter, we're going to cover the actual steps or stages of that process. So we're going to start off with talking about interphase and the sub-stages that are contained within interphase. And then we'll briefly talk about G0. And then we will go through the phases of mitosis, followed by cytokinesis. So that's where we're going. Okay, so make sure you can see that. Before we start diving into mitosis, we have to talk about the cell cycle in general. Okay, so the cell cycle, remember that these are the steps that occur in a parent cell leading up to the eventual cell division and the production of two identical cells. Those are called daughter cells. Okay, so you can see in this cell cycle, this is pretty neat. It actually shows the relative lengths of time for each of these phases. So it's a cycle, so it's a circle, and it proceeds in the clockwise direction. So we can see those arrows. And you'll notice, I'm not sure if you can see my little highlighter, so let me make sure I have I'll use my highlighter here. Okay, so you'll notice the cell cycle actually starts right here. And it's going to continue, again, in this direction, all the way around until we get back to here. When we get back to here, we would have two cells. Okay, now this starting point here is the beginning of the first phase, which is called interphase. That's the first phase of the cell cycle, and that's going to precede mitosis. Okay, notice how long interphase is. It's going to be composed of G1, S phase, and G2. So we say if we're looking at the cell cycle time-wise, interphase is going to be the longest phase. And that's going to be followed by mitosis, which ends in cytokinesis. Okay, and we're going to talk about mitosis and cytokinesis as separate events, but you should really understand that they kind of occur simultaneously. So cytokinesis is going to occur really during the end stages of mitosis. And again, at the very end here, we have two identical daughter cells. Okay, so we have interphase, which is the bulk of the cell cycle, followed by the mitotic phase, okay, which is much shorter. And the mitotic phase consists of mitosis, which we're going to learn is also called karyokinesis. And what that is the division of the nucleus. And then that is followed by cytokinesis. That's the division of the rest of the cell. Those together collectively are called the mitotic phase. So mitosis and cytokinesis make up the mitotic phase. Okay, so let's dig a little bit deeper. And again, we're going to start off, threw this in there. So this is a simplified picture of the cell cycle, again, showing timing. And instead of having a lot of words like what was on the last slide, it's really, really simplified. And we've got some nice colors here to show the different main phases. Okay, so everything in blue. So we got G1, S, and G2. Those are all part of interphase, again, preceding mitosis, where we have M phase up here, or the mitotic phase, which includes mitosis and cytokinesis, shown in red. Okay, so again, you can see that that's usually pretty quick compared to interphase. The length of time each of these phases depends on the species. Okay, some organisms can go through the cell cycle quickly, like bacteria, like E. coli, they can duplicate in like 20 minutes. But other organisms, their cells take longer. Okay, and again, we are going to talk about G0 in just a minute. Okay, let me see if I left anything else out. Okay, so I just gave you kind of an overview. So again, in blue here, we have interphase. Interphase is composed of G1, which is also called GAP1, S phase, which is also called the DNA synthesis phase, so that S stands for synthesis, and then G2, which stands for GAP2. And we're going to go over each of these stages, but just as a summary, interphase is a phase of normal growth and preparation for cellular division. The one key thing that happens here, it happens during S phase, the second phase of interphase, is DNA replication. That is key to this process. And then again, in red, we've got our mitotic phase composed of mitosis and cytokinesis. This is where the genetic material, those chromosomes, that DNA, gets divvied up between the two nuclei that are going to be produced, and then the rest of the cell material also gets distributed. And then we end up with two identical daughter cells, exactly the same genetic information as each other and the parent cell. Okay, so for humans, let's say this is mitosis happening in a human somatic cell, a human body cell. We would go from a cell containing, one cell containing 46 chromosomes to two cells. And each of those cells would both contain 46 chromosomes. They would contain the same 46 chromosomes as each other. And if we compared back to that original parent cell, it would be the same 46 chromosomes, right? Those chromosomes were replicated, and then those sets of chromosomes were then split. Okay, we end up with 46 chromosomes in each cell, and again, the cells are genetically identical. Okay, so again, let's start off with interphase. This is going to come before mitosis. It's everything in blue here on our picture. And again, during interphase, we've got at least one really important step, but we've also got some normal growth happening. Okay, so we're going to go through G1, S, and G2. So G1, which comes first, it's called the first gap. Okay, gap is kind of misleading. It makes you kind of think of rest, but the cells definitely aren't resting. Okay, instead they're getting ready for DNA replication. Okay, if you were to take a look at an interphase cell under the microscope, you don't see differences, right? The cytoplasm looks pretty much the same. The nuclear envelope is still intact. There's still a nucleus. The DNA doesn't look much different, right? But if we were to look really close biochemically, we would see that the cell has amped up its enzyme production. It's starting to produce more of the proteins that are involved in DNA replication, more of the proteins that are involved in chromosomal packing or packaging. So it's definitely biochemically active. Okay. Okay, that's G1. Now the cell will then enter into S phase. And again, that S stands for synthesis. So that means something's being made or synthesized. What is that? It's DNA. So the entire genome of the cell needs to be copied so that we can go from one cell to two cells with exactly the same genetic information. That's going to happen during S phase. So during S phase, the DNA is replicated. So if we were to take a look inside of that cell, and I'm going to draw a chromatid. Okay, so the DNA is not this condensed, but just for the sake of being able to actually see it, we would go from 46 of these one-armed chromosomes, or monad chromatids, monad meaning one arm, okay, and the chromatid is an arm of a chromosome. Once this gets replicated, this is what it looks like. There's one copy, that's that one arm, and here is its copy. Okay, so we go from a monad, shown here, to a dyad chromosome, shown here. So di meaning two arms. Okay, those arms, this is one arm, right? This is the second arm. They are exactly identical to each other. So yes, they're the same size, they're the same shape, they have the same location of their genes, and the same exact alleles. There's no variation here. Okay, if chromosome arm number 1 contains the gene for eye color that codes for blue eyes, well, you're going to find that very same allele on sister chromatid number 2 because they are exactly identical. Okay, one was made as a copy of the other. These arms are held together. Let's see if I can change my color here. Right here in the middle, see where they're connected? There's going to be proteins in there, which are going to hold the two arms together there in the middle. And that region, that cinched region, is called the centromere. Okay, that's the region of the dyad chromosome where the two sister chromatids are held together. Okay. We say by the end of S phase that all of the genetic material has been copied. So in human cells, we would go from 46 monad chromosomes, or we could say monad chromatids, that's the same thing, to 46 dyad chromosomes. Okay? What else happens during S phase? These structures called the centrosome, they're going to get duplicated. Okay? So the centrosome is going to be a structure which is going to be responsible for eventually pulling the two arms, right? So arm 1 and arm 2 of each of the 46 chromosomes if we're talking about humans. The centrosome is going to be a structure that's responsible for pulling those two arms apart. Because that's what we're going to do. We're going to take that replicated chromosome, right, with two identical arms or sister chromatids, and we're going to pull those arms apart and put them in opposite cells. That's going to make those cells identical. Okay, so again, those structures are called centrosomes. They're going to be, there's going to be 2 of them after they get duplicated, and they're going to move to the poles or opposite sides of the cell eventually. And something called the mitotic spindle is going to form out of those structures. And those spindle fibers are going to be what actually does the pulling. Okay, we're going to get there in a little bit. For now, you just need to know that the single centrosome present in the parent cell gets duplicated. And so now there's going to be 2 of those centrosomes by the end of S phase. Okay, those are the highlights of S phase. During G2, we're still in interphase. This is all still before mitosis. G2 stands for the second gap. And here the cell is going to prepare for mitosis. So anything that hasn't been replicated, any organelle like mitochondria, or if it's plant cell chloroplasts, those kinds of structures are going to be copied. Ribosomes, things like that, those are going to get copied. The cell is going to need energy for mitosis, and so we're going to have cellular respiration, which we haven't talked about yet, but we're going to have that going on to make plenty of energy that's going to be required for this process. And lastly, the cytoskeleton, which we did talk about already, that's going to be broken down. So a lot of the elements of the cytoskeleton are going to be required for mitosis, for the actual pulling of those chromosomes apart. And so the cytoskeleton in general is going to start to break down and gear up for mitosis. Okay, so that's interphase in a nutshell. Key things that happen is cells gear up for mitosis, DNA replicates, centrosome replicates. That's pretty much it. Okay, I need to briefly take a sidestep here and talk about G0. G0 is not part of mitosis. In fact, G0, which is right here, it's really within G1, so it's really before the cell starts to gear up for division, basically because a cell that's in G0 is going to be a non-dividing cell. Okay, we use the term quiescent. It's a quiescent stage, which just means non-dividing stage. Some cells enter this when, environmental cues are present, and then they'll come out of it when the environment is more favorable to division. And then there are some cells that will enter G0 permanently. Okay? You have some cells in your body that really rarely will divide, especially once you reach adulthood. Some examples of some quiescent G0 cells, your nervous tissue cells. So neurons, they tend not to divide, although there is some evidence that there is some division that happens. But in general, they're going to be quiescent G0 cells. Okay, so again, some cells never enter that phase. For example, your epidermal cells, there's constant turnover there of your skin. So it depends on where we're looking, what cell type we're looking at. Okay, so that's G0. Okay, we talked about interphase, we talked about G0. We're going to now talk about the mitotic phase. Okay, so the mitotic phase is composed of mitosis and cytokinesis. And again, remember at the end of this process, we end up with two daughter cells that are identical to each other and to the original parent cell. Okay, mitosis is also called karyokinesis. So karyokinesis, mitosis, those terms are interchangeable. Okay, mitosis is considered to be the division of the nucleus. So during mitosis, we go from one cell, the parent cell, with one nucleus, and by the end of mitosis, we have two nuclei. And those nuclei, again, are genetically identical. They have the same number and types of chromosomes. Okay, so again, if this was a human cell, we would go from one nucleus with 46 chromosomes, if we want to be specific, 46 monad chromosomes. And by the end of mitosis, we would have two nuclei that each have 46 chromosomes. Again, if we want to be specific, those chromosomes would be monad chromosomes. Okay, remember the difference between a monad and a dyad? Let's see if I could do this. That's a monad. That's a dyad. Okay, I didn't draw that arm long enough. Okay, so a monad is a one-armed chromosome, a dyad is a two-armed chromosome. The only time that you get those X-shaped chromosomes that you're probably more familiar with seeing is during cell division, during mitosis and meiosis, when genetic material is being divided or divvied up. Okay, the rest of the time, your chromosomes are monads, and they're not condensed either. They're more diffuse. Okay, so we've got mitosis or karyokinesis. That's where we go from one nucleus to two identical nuclei, and that's going to be followed up by cytokinesis, where we go from one cell to two daughter cells. Okay, one cell with one nucleus to two daughter cells. Each of them have their own nucleus. All right, so let's dive deep. We're going to start with mitosis, which again we can call karyokinesis, and there are stages here. We've got prophase, and then your textbook likes to talk about prometaphase, okay, which we'll cover that as well. In lab, we kind of leave that out and we just stick to the main phases. Prometaphase is kind of a subphase. So we got prophase, prometaphase, which means before metaphase. And then we've got metaphase, anaphase, and telophase, okay? Let's start with prophase. Okay, so during prophase, this is what's going to happen. The nuclear envelope, remember that double membrane structure that surrounds the nucleus that houses the genetic material, that's going to start to break down or dissolve, okay? The dark spots in the nucleus, called the nucleoli or the nucleolus if there's just one of them, remember that was the site of ribosome assembly. Those darkly stained regions found in the nucleus, those are going to also disappear. Okay? What else happens? The centrosomes that were duplicated during S phase of interphase, those are now going to spread out to opposite sides of the cell, which we call the poles. Okay, the poles of the cell. You can kind of think like North Pole and South Pole of the Earth. They're on opposite sides of the planet. And that's how we're using the term pole here as well. So when we say centrosome move towards the poles, that means opposite sides of the cell. Okay? Now in this image here, from your textbook, you can see this structure and this structure. Those are the centrosomes. Now this is an animal cell. How do I know that? Well, there's a couple of things that tell me. Number one, I don't see a cell wall. I only see a plasma membrane. Okay, and number 2, if I look at the centrosomes, I see those orange things in the middle. Do you see those? Right here and right here. It's a doublet, a set of 2 of those orange things. Those are called centrioles. Those are present in animals and not plants. Okay, you got to remember back to, I think it was chapter 4, when we talked about those structures. Okay, they're present in animals but not plants. Let me erase that so you can really see those structures. Okay, so it's a doublet or a set of 2 centrioles in the center of the centrosome. They're kind of organizing. Let's look a little bit further at the centrosome. So what are the blue things kind of radiating out from the center? Those are microtubules. So remember, those are elements of the cytoskeleton. They're made up of the proteins alpha and beta tubulin. Alpha and beta tubulin can be combined, right, using energy. And we can actually make these spindles longer or shorter. So microtubule fibers can grow and they can shorten. And that's going to be key in division here. Okay. That's the next one down. It says microtubules start to form the mitotic spindle. So again, that's this blue stuff coming out. That's going to make the mitotic spindle. And lastly, our chromosomes condense. Okay, they're going to be packaged. We talk about how they were packaged in the beginning of this chapter, right? So we've got our histone proteins that form nucleosomes, the beads on the string, and then those are going to be put together using more proteins, eventually becoming thicker and thicker and thicker to form dyad chromosomes. And so found here in the nucleus, okay, you can see this dotted envelope, right? That's the nuclear envelope breaking down. And you can see that we have 1, 2, 3, 4 dyad chromosomes inside that nucleus. Remember, dyad because they have 2 arms. Okay? So this cell started off with 4 monad chromosomes. Now we have 4 dyad chromosomes. Can you guess how many chromosomes each daughter cell will have? We just jumped to the very end of mitosis. We're going to produce 2 daughter cells from this one parent. And if we were to look inside those 2 daughter cells, they would each have 4 chromosomes. We would say monad chromosomes. Okay, so chromosome number in mitosis stays the same. The parent cell right here that we're looking at has 4 chromosomes, and each daughter cell will also have 4 chromosomes. Okay? Okay, so that's prophase. The next stage here, again, is prometaphase. So the events from prophase continue. What does that mean? So the rest of the nuclear envelope, if it hadn't already broken down, it breaks down. The nucleolus, again, if it hadn't finished disappearing, it would disappear. Those centrosomes now completely migrate to opposite sides of the cell. The microtubules that are growing out, you can see these, right? These blue strands, these blue fibers, I should say. These are part of the mitotic spindle. Okay, or these are the spindle fibers. They're going to grow out from each side. So here's one pole, and here's the other pole. And as those mitotic spindles form, they're going to reach out and grab on to each chromosome at a structure called the kinetochore. So you can see this term here, kinetochore. This is in the centromere region of the chromosome, and there's going to be proteins here. Okay, so right here is the kinetochore. And these proteins are going to allow the mitotic spindle microtubules to attach. Okay, and the spindles are going to attach on both sides. So here, there are spindles attaching to this side of the kinetochore, and the same thing is going to happen over here. Eventually, let me grab my pen. Eventually, what's going to happen, you can see, is the chromosome here is going to be split right down the middle. And so the sister chromatids, one of them is going to be pulled that way, and the other one is going to be pulled that way. So again, what are we doing here? We're taking the genetic information and we're splitting it up. These are identical copies of each other. And so we're putting one copy in one cell and the other copy in the other cell. Okay, eventually. We're not there yet. So pro-metaphase, the mitotic spindle is going to attach to the kinetochore on each side of the chromosome. Okay? We're setting everything up. Okay, so pro-metaphase is followed by metaphase. And metaphase is pretty simple. When you think of metaphase, I want you to think middle. Okay? So those chromosomes, those dyad chromosomes, are going to line up now in the middle of the cell. And the middle of the cell, depending on what textbook you're reading, is called all different kinds of things. So for example, some scientists will just call it the middle of the cell. Some will call it the metaphase plate. And some will call it the equatorial plate or the equatorial plane. Okay? Just means the middle of the cell. So they line up there in single file order. And again, the mitotic spindle is going to be attached on both sides of each chromosome. So here we've got 1, 2, 3, 4 chromosomes lined up. Okay, and again, each chromosome is going to be attached at the kinetochore to the mitotic spindle of both centrosomes. So here's one, and here's the other. Okay, so now they're perfectly lined up. This is neat. Here's a picture from your textbook. I haven't been pointing this out. This is a fluorescence micrograph. So this was taken with a fluorescent microscope, showing you in blue the chromosomes lined up, and in green, green is lighting up any microtubule. So the mitotic spindle is lit up in green. So it's showing you how those chromosomes line up and how that mitotic spindle is going to attach. It's a pretty cool picture. Okay. Notice how the sister chromatids are still attached to each other at the centromere. Okay, so now we're going to move from them lining up to now sister chromatids are going to be pulled apart. And so those cohesin proteins that were holding them together, they're going to break down and allow those sister chromatids to be pulled in opposite directions. So again, what are we doing here? We're dividing the DNA, right? We're pulling it to opposite sides of the cell. So if you have a dyad chromosome, that's going to be split, and then each arm is going to be pulled in different directions. So we say that sister chromatids are pulled apart during this step. And again, that's called anaphase. Sometimes I'll use anaphase away, okay, for that. And we can see that happening here again in this fluorescence micrograph. You can see genetic material being separated. Okay, the genetic material in this case is this bluish purple. Okay, and again, the microtubules being organized by the centrosome are what's doing the actual pulling of those chromosomes apart. What's happening here at the base, the center I should say, of the centrosome is these mitotic spindles are being shortened. So alpha and beta tubulin are being removed from this end. So there's literally a pulling force of those sister chromatids towards the opposite poles. Okay, so anaphase away. Last phase of mitosis before we get to cytokinesis is called telophase. And so telophase is kind of the opposite of prophase. What's going to happen here is chromosomes now have completely reached the opposite poles or sides of the cell. These chromosomes are in the monad form or chromatid form, right? Single one-armed chromosomes. What's going to happen now is they're going to start to decondense a little bit. Instead of being tightly coiled chromosomes, now they're going to become less tightly coiled. They might even go into the beads on a string form. Okay, so they start to decondense. The nuclear envelope will reform around those regions of genetic material. And so we're going to have two nuclei, okay, both surrounded by a nuclear envelope. They're going to form. What else happens? That mitotic spindle will now start to break down. And lastly, we have cytokinesis. So during cytokinesis, the cells will actually split. Okay, and so what happens here is, well, the cytoplasm gets divvied up, and the cytoplasmic organelles like the Golgi and ribosomes, the centrosomes, mitochondria, lysosomes, all of those structures, chloroplasts if we're talking about plants, they're all going to get divvied up between these two cells so that there's generally an equal number of organelles and cytoplasm in each of those cells. And then the cells actually physically split, and in the end we have two genetically identical daughter cells. Now, before I jump there is going to be a difference between plant and animal cells as far as how this process happens. And that's pretty much because there is a difference in plant and animal cells structurally, right? Plant cells have an extra layer, the cell wall, that they have to contend with. So let's take a peek here. So again, in animal cells, there isn't a cell wall. And so what's going to happen in animal cells is we have elements of the cytoskeleton, specifically actin, okay? They're going to come to kind of the central region between the two nuclei, and they're going to form what's called a contractile ring, which is going to literally pull the plasma membrane, kind of like cinch it or pinch it in. And one of the key hallmark things that we see under the microscope when this is occurring is this indentation right here, which scientists have coined the cleavage furrow. Okay, that's the start of this division of the cell. And eventually, once this pinches and pinches and pinches, we end up with two cells. So a key hallmark of cytokinesis in animal cells is the presence of the cleavage furrow. We don't see that in plant cells. Instead, in plant cells, what has to happen after we have our two nuclei, or while they're forming, really, this process of cytokinesis is really happening while telophase is occurring. Okay, so usually. So what's going to happen in plant cells is, we can't just pinch apart the two plasma membranes because we have a cell wall. So what will happen are these vesicles carrying cell wall components, as well as plasma membrane components. They'll start to be sent to the midline, the middle of the cell, and they'll start to lay down those components. And so early on in cytokinesis, what we'll see in plants is something called the cell plate. And that's this line that starts to form between those two nuclei. And that line starts to extend and extend and extend. And eventually, by the end of cytokinesis, we have two distinct cells, with their own plasma membranes and their own cell wall components. Okay? So again, the steps and the sequence of events that happen in both plant and animal cells for mitosis are the same. The only difference is what cytokinesis really looks like. Okay? And again, also, animal cells have centrioles at the center of their centrosome, while plant cells do not. Okay, your turn. I'll take a break from talking, and you're going to go ahead and you're going to finish the Are You With Me, the second last Are You With Me? So you're going to define these terms, and then you're going to go ahead and you're going to answer these questions, which involves a lot of sketching. So you're going to want to make sure, again, that you make nice detailed sketches, use those colored pencils, and then you're going to take pictures or scan, however you want to do that, and you're going to upload them right to Blackboard. When you're finished, come back because I have a couple questions for you. Okay, so here's the first question. Identical copies of chromatin held together by cohesin proteins at the centromere are called histones? Are they called sister chromatids? Are they called nucleosomes? Or are they called chromatin? Okay. So think about this one. Where's my pointer? Okay. So identical copies of chromatin held together by cohesin proteins at the centromere. You should automatically start to picture our X-shaped structures. Okay, and again, held together in the middle there would be the centromere, and there would be those cohesin proteins located there holding the two arms or sister chromatid of the dyad chromosome together. Okay, so there's your answer there in blue. Those identical copies are called sister chromatids. Okay, next question. Which of the following events does not occur during some stage of interphase? So we're talking about interphase. DNA replication, organelle duplication, increase in cell size, or separation of sister chromatids. Which one does not happen during interphase? Okay, so hopefully you answered that orangish yellow, separation of sister chromatids. That doesn't happen now. When does that happen? Hopefully you answered anaphase, right? Anaphase is the phase where sister chromatids are pulled apart, where those cohesin proteins are broken down and they can actually be pulled apart. The rest of those are correct. Remember, DNA replication is going to happen during S phase of interphase. Organelle replication happens during G phase, usually G2 phase. Increase in cell size, I'm not sure if I touched on that. So we talked about growth, which hopefully you know that growth equals an increase in size here. That's going to happen in G1, maybe a little bit in G2 as well. Okay, next question. These are the last two. So first one here. Chromosomes are duplicated during what stage of the cell cycle? Are chromosomes duplicated during G1, S, prophase, or G2? Remember that term duplicated is the same as replicated. We can use those interchangeably. So we could say chromosome duplication or chromosome replication. When does that occur? Hopefully you answered S phase. So S means synthesis of genetic material, right? That's going to happen during S phase of interphase. Last question. Separation of sister chromatids is a characteristic of which stage of mitosis? Does separation of sister chromatids happen during prophase, prophase, metaphase, anaphase, or telophase? And hopefully you answered anaphase. Okay, remember anaphase away. That's the last slide. Okay, so that's it for chapter 10. You've got to finish your Are You With Me's and get those in. And then you've got chapter 11, and then you'll have the chapter 10 and 11 quiz. It's a joint quiz this week.